Hello everyone! Today is November 25th and we are going to talk about bioaerosol sampling with the SKC Biosampler with Chloe. Hello Chloe, how are you doing?
Good. My name is Chloe Wooldridge and I got my Bachelor of Science in Civil Engineering from the University of Kansas. And now I'm here at UT working on my Master's. I'm going to be telling you guys about the operation of the SKC Biosampler impinger, which is this device right here. This is used to collect bioaerosols, and bioaerosols are any airborne particle that contain biological material like bacteria, fungi, viruses, and you can collect those into a liquid and then use that liquid to extract DNA, and then you can sequence that DNA and see what kind of bacterial, fungal species you have present in your sample.
Very nice Chloe, very nice introduction. So, what are you going to do?
Now I'm going to talk to you guys about how to actually operate the biosampler. So to do this, you need to use a pump along with the biosampler.
Alright.
And we're going to connect the pump to the outlet nozzle of the biosampler, and then that's going to force air to be pulled in through the inlet nozzle. And that air is going to be sucked into where we have the collection media, which I'll pour into this in a second. And it's going to create a vortex that'll gently collect bioaerosol particles into the liquid, and with the intent of keeping as many of the cells alive as possible in case you want to do plate culturing or something like that. And then we will pour the sample into a sterile vial. So step by step, how to operate the impinger. We're first going to want to turn on a flame to keep the environment sterile around the impinger. And I'm going to provide gas to the Bunsen burner and then ignite it. And then we're going to pour 20 mils of our chosen collection media into the bottom portion of the impinger, and we use phosphate buffered saline, which is commonly referred to as PBS. So you're going to want to remove the top two parts of the SKC biosampler and then pour your collection media into the bottom. Then you replace the top two pieces, as so. And then we're going to hook this up to our pump, which has been pre-calibrated to 12.5 liters per minute for a flow rate. You're going to put the inlet to the pump on the outlet of the biosampler, like this. And then we turn the pump on, which creates a vortex in the liquid like I mentioned earlier. And then you can sample with this device for up to 30 minutes is recommended in the protocol, but we've sampled for longer. And I'll post 2 papers that talk about, or that use the biosampler, so you can look at the methods that they use for how long they run it, what kind of pump they use, etc.
But Chloe, I have a question. How is exactly this working?
Yeah, so this pump is pulling air out from this nozzle of the impinger, which in turn is causing air to be pulled into this top nozzle.
Okay.
So air is coming in through here and then getting sucked up out through here. And that's what's creating the vortex inside of the impinger.
Because of the special shape, right? They have a special shape, right?
Yeah, so the air coming in through this nozzle is being pushed out through these little feet that you can see sticking out in here.
Alright.
And then getting pulled up in the middle of all the feet, there's a hole where the air is getting pulled back up and then going through the pump.
Right.
So next we want to isolate our sample from the impinger. So after you finish sampling for whatever collection period you've chosen, you turn off the pump. You disconnect the pump from the impinger. And again, we're doing this under sterile environments so as not to contaminate the sample. And then we're going to pour the 20 ml sample that we've collected into a sterile tube that has been nicely labeled with what kind of sample it is, my initials, and the date. So you're going to remove the first two portions of the biosampler. And I'm going to set these down since we're not worried about contaminating these anymore since we're done sampling. And then you're going to pour the sample into your sterile vial. So that's how you use and operate the SKC Biosampler. Thanks for listening, and make sure you guys follow us on Twitter at UTBio.